So today I will talk about reconstructing dimensional motor unit control using models and targeted neural implants. So you probably all know about the motor unit, yeah, but just briefly, you have in the ventral horn of the spinal cord alpha motor neurons. So the alpha motor neurons with its body, the axon, and the innervated the muscle fiber to its axon forms the motor unit. Yeah? And this is the last, it's a functional element. Yeah? By many has been described as the last quantum of muscle actions. Yeah? This is the last pathway of movement. That when there is an action potential generated here, you will always have motor unit twitch, a force. In healthy people, when you have a healthy neuromuscular junction. And we don't have many motor units. So, for example, this muscle, the first dorsal interosseous muscle, that mainly controls the abduction and flexion of the index, has 100 motor units. So the number is quite small if you think about it, especially from a neuroscience perspective. Well, in motor cortex only, you have 1 billion of neurons. So the dimension is very significantly different. So only few of these neurons make us move, and move quite well as well, because we can exploit a large number of degrees of freedom. So we know a lot about motor unit because of pioneering works about EMG. And we know now a lot also about EMG. And basically, when you have, in this case, is an example from a cortical volley that reaches the spinal motor neurons. But this can also be an afferent volley from, for example, the afferent muscle fiber in the intracusal muscle fiber of the skeletal muscle. You have a volleys in the axons that reach the muscle fiber, okay? And then here, for example, in this case, we have an implant inside the muscle, and this implant is recording, as you see here, an interference EMG signal, okay? So this interference EMG signal is an algebraic summation of the motor unit action potential that are identified, plus some added nodes. So in other words, if you identify these, you can always reconstruct that. I've been very interested, and what the field has been very interested in, is to decompose this signal, so to remove the constituent motor unit action potential, so we can extract the neural tone of movement. So if you go from this signal to this signal, you basically have the effective neural drive to the muscle, which is the signal that makes us move. And if you have this signal, then you have a muscle that is filtering this signal, okay? But if you are interested in robots, that is the best signal you can get, because that is really a direct tap to the neural code of movement. So, and with this signal, we can now study quite well how the brain and the nervous system is coordinating movement. Because for many, many years, we have been linked to a signal that is gross, that is crude, that is not accurate of the neural information. And one problem, in my opinion, that is still quite relevant, is how the central nervous system organizes the control of multiple muscles. And one model that I find very interesting is to compare the lower limb muscle with the hand muscle. Why this is relevant? This is relevant for two reasons. The first reason is that when the vasti muscle, so these are synergistic muscle in the leg that control the extension of the leg, so they do this movement. So the vasti muscle are connected to the bones, to the patellar tendon. They share the same tendon, okay? So in this biomechanical, you have to imagine that they project forces in the same direction. And they also receive input from the Golgi tendon organs that is located there. On the other hand, the hand has individual tendons that control each digit individually. Okay, so we can move each digit individually, but we can also move it synergistically, for example, during a pinching. So comparing such tasks, so you perform a synergistic task. In this case, it's a constrained task, right? Because in theory, you cannot control this muscle individually, yeah? Because you would always move the leg in that direction. But here you can know that you're moving in that direction. So we were very interested to compare these two muscles. Why we're interested? Because there is in the literature, if you take a look at it, for the last 40 years, the main concept is that the vast muscle receive one common synaptic input. So that from a neural perspective, although we have two anatomical different muscles, from a physiological and neurological perspective, the common input, the brain sees these two muscles as one. And I wanted to verify, is this really true that from a neural perspective, these are really one muscle? To me, it sounds quite strange, because if you think about it, these are two different muscles, although they're projecting the same. So why did nature create two muscles? Yeah, and if you look at evolution, so if you look at any, if you look at the evolution, it's one of the most economical things that exists. Evolution, everything that does not make sense from an economical perspective is cut off. So the evolution doesn't like to waste energy. And we know this across many, that we have so much proof of that. So the idea was, do we really have one common synaptic input? So now with the technology that we have, with looking at motor units, we can test this hypothesis very, very carefully. And so what we did, we designed a study where we were performing synergistic movement with the tenor and the first dorsal interosseous muscle, so these two muscles here that control pinching. And then we had we find the motor unit from this muscle. So you see here in this color, in purple you see from the thumb, and in green from the first dorsal interosseous. And these are two motor units from that muscle. These are two motor units from the other muscle. And if you plot these two motor units, you can see that they share the same manifold. So they are very correlated. When we look at the vacuum muscle, we found something quite surprising. So that there are some motor units from the same muscle. So in this case, this is the vastus medialis, and it's the vastus lateralis muscle, that have either very strong common input, so they are very correlated. And some other motor units, actually, they do not lie in that manifold, but they are from that muscle. So they don't receive the same input. And so then we asked, what is the proportion of this unit? What is the distribution? There are multiple ways of checking that. So you can imagine here you have hundreds of motor units, and we can apply simple machine learning methods, for example, factor analysis or principal component analysis, that can estimate the amount of motor units that they are in this cluster or in this cluster. So basically, what are the time varying inputs? And when we looked at that, I will not go into details. However, factor analysis was very efficient in identifying these inputs. And for the head, as we can imagine, the input are very muscle specific. So this input here, so basically this is like so imagine that the factor analysis or PCA. Also, non-negative multifactorization gives you two outputs. One output is the coefficient. So the coefficient tells you the correlation between that signal to an estimated component. Yeah? The component that you estimate has the same length, is a vector with the same length as the discharge time of the motor. But you are reducing the dimension in time. When you take this signal, which is the signal that is reconstructed and has the same dimension of motor unit in time, you can correlate this signal to the individual motor unit. And this is a very important step, because you don't know what the PCA or factor analysis is doing. Even if you know the math, you need to understand what is the relation between that signal and the motor unit, because what we care is how we reconstruct the motor unit variance. That's what we care from a physiological perspective. And when we do that, you can see that this is, so each of these plots is basically the correlation, in this case of one motor unit. These are across all participants of that motor unit with the latent component from the factor analysis. And you can see that for all motor units, we have a very high correlation, almost one with the latent component. So what factor analysis identified is physiologically significant. And however, we don't have any unit here in this plot. If you would have some unit here, it means that it will be correlated with both components. But as you can see, no, they lie on an orthogonal plane. So this means that the input are not correlated between these two muscles, even though the task is synergistic, okay? That they are doing a pinching action. On the other hand, in the leg, you can see that there is still a very strong unique component. In the VM, this is very, very high, but there is quite a lot of motor units share the same input. So basically, there are these motor units here. While these motor units here are either in the muscle specific, but what we were looking at, and this was an unexpected finding, was that there are so many motor units that are responding to the input from the other muscle. So you have a muscle-specific input, then you have an input, the two inputs form an algebraic summation, and then some other motor unit responds to the input of the other muscle. So there is a synergy, but it's very complex, this synergy, as you can see. So then, after looking at this finding, there were two important questions that came to mind. So the first one is, if we have these two inputs, can this dimension be individually controlled? So if we give human now the possibility to control this neural signal in real time, can we actually split these two muscles? And the second question, what are the dimensions in the hand? I mean, if this is a synergistic task and there are no synergy there, what are actually the number of dimensions? So I will first now go through this and then that, but so you will have both in the next slide. But what I wanted so these questions, they are very neuroscience applied. However, if some of you has a background in neurorehabilitation, you know that there are so many pathologies. So for example, just to name a very simple one is patellar dysfunction. Many people suffer from the patellar that is popping out the knee and is not stable, the knee. And what physiotherapists try to do is try to selectively strengthen the vastus medialis. But how they do it, they do it a bit like, by estimating this. They don't have a direct access, so we don't know how to actually train on the vastus medialis. So if we can develop a training intervention with the neurofeedback, and that would work, it would really help physiotherapists in developing new neurorehabilitation intervention. So what we did to understand this, because we'll not go through the details, but there's a lot of crosstalk. So if you put here the surface EMG over the muscles, you will find that there is a lot of crosstalk. So it's very hard to selectively be like 100% sure that the activity from one muscle is coming exactly from that muscle. There are techniques to do offline that you can remove crosstalk offline very well, but not in real time. So what we did in this case, we implanted several intramuscular sensors across all the direction of the vastus lateralis and the vastus medialis. So, and specifically, the hypothesis there that I think if what we were thinking is you cannot implant so many because this is hard. So we wanted to really target some specific portions, and we were thinking that the best way to target this specific portion is if there must be a change, it must be like proximal to distal, because where the fascicles are very far apart of the muscle fiber, so that the motor unit nervation zones are different, okay? And also here. And then with ultrasound, we confirmed that these were the muscles that we wanted to investigate. And then this is the experimental setup. That we implanted 7 of these sensors and together with the surface imaging. You can see here that the surface imaging grid there was a cut because here was where we implanted the electrode, so we have a very similar task. And then we designed a neurofeedback. This is a simple neurofeedback, but it's very effective because you can take the implant data and then create a Cartesian axis, Cartesian plot, and you can scale both the activity of the vastus medialis, vastus lateralis, either in a time signal or with like one muscle against the other. So participant can either go with, can control in real time this cursor in this zone. If it goes in this zone, it means that the vastus lateralis is mainly controlled. And if they go in this zone, it is the vastus medialis. But as you can imagine, there are so many motor units, so many vectors, and so we had to make a training section. This lasted around 30 minutes for every person. You can see here an example of when of the signal and of the decomposition, so to find the motor unit. So the first thing was, so now we have so many implants. So this is you will see this plot so that you have different colors. These are color-coded each implants in the data. So the first was to check whether you have a correlation when you do a force task. And that's what we always be doing. Like for the last 10 years, 20 years, we always have force feedback. And that's okay to do force feedback because you are isometric force feedback because it's very controlled. However, if you do this, you find a synergy, obviously, because they're both electric sensors, and you can see here very well that each of these electrodes is basically the same, and you can assume that there is a synergy, that there is a strong correlation, because these are two masks we do leg extension, and this is what has been done. But what about now if we train subjects not to do this? And with the EMG, so these are just visual feedback. You can see after some training, some of these electrodes, they were completely not correlated with the other signal. However, you can already note here, these are two different subjects. You can see that this electrode here, in the proximal VM, can be silenced when the other are activated, and can be activated when the other are silenced. We'll go back to this pattern. What we found across all subjects was that the proximal VM was the only part, after a few minutes of training, that could be, this was in 9 subjects, all of them showed that the proximal VM could be independently controlled from the vastus lateralis and the distal vastus medialis. So you can see this is the distal vastus medialis, and this is the vastus lateralis. You can see that there is a very strong, these are the motor unit, and you can see that there is a very strong correlation. However, the proximal VM can independently control. When we look at this, so this is the force feedback, and this is the RMS feedback, so this is the EMG feedback. You can see that all subjects, each of these is one subject. During force feedback, whenever you compute a correlation from all these electrodes, or this, you find a high probability. Okay, so you find a synergy. On the other hand, here, when you use RMS feedback, you can see from this side to this side, that there is really no correlation, and it goes significantly high when you go here. So this means that the number of dimensions is bigger, and is also anatomically constrained. And here you can see very clearly what happened from the composition. So if you decompose the intramuscular signal, you see here the raw EMG and the spike trains. These are two. These are, for example, during a trial, one trial and another trial, where they were trying to split the activity. You can see that during one trial, there are these two motor units and these two motor unit active, and another trial where there was more emphasis on the vastus medialis, you can see that this signal is completely flat. We have no any more motor units, but we have more motor units. So there is a strong neural drive to that muscle. And this is the opposite case in this case. You see also that you have here it's more silent, here it's more silent. So it is possible to switch motor unit from these two synergistic muscles. So this means that from a real-time perspective, we can now train this muscle selectively. And the dimensions are not fixed. We have multiple dimensions. So in the serogenic vasti muscle, it seems that voluntary dissociation of the neural drive is possible, but location specific. So this is a speculation because we recorded 10 subjects. So 3 of these subjects managed to split completely. And these 3 subjects were 3 subjects that go to the gym, that train a lot at the gym. So this is, of course, it's not a statistical high number, but I just wanted to share with you that it could be a lot of this. So with training, with a lot of training, people may be able to selectively activate this muscle, or it could be also an innate ability. And I find it's quite interesting that this was a 30 minutes of training. It was a very short session, but maybe with training now we can augment the number of dimensions that we can extract from a constrained muscle. So now this brings to the other question. What are the dimensions in the hand? So this is it must be very high here. So what we did, we developed first we wanted to record the dimensions, and the dimension now we can record with AI very well. So it's a very good representation of movement. You can estimate the kinematics of the human hand with residual neural network. With so many neural network now you can estimate position. And we basically recorded all possible tasks. that the hand can do. So from synergistic movement, equilibrium movement, and so on. And then we decompose the signal, and for each of these signals we decompose, and we have some spike trains for each of these digits. And what was the finding here, which was quite expected, but it was nice to demonstrate it, is that, for example, during a thumb fast movement, a ring fast movement and a pinky fast movement, on the same row, you have the same motor unit, okay? So this is the same motor unit, for example. This is the same motor unit that was tracked on thumb, ring, and pinky. And you can see, you can see that during the thumb fast movement, this motor unit is flat. On the other hand, when the ring fast movement, this motor unit starts encoding the movement very well. And then here it's silent. And so motor units are really tonically discharging. And so motor are like always phase because of this movement, it is a phasic movement. And you can see here also in the pinky fast. So there are some motor units that we define them as a prime mover. So there are motor units that construct the movement. And so motor units that hold the posture because the hand is still in this way. So there must be a posture to be controlled. Because if you would relax the hand, it would go like that. So there must be some motor units that are encoding the posture of the hand, and these are the motor units. Then, this shows you a little bit more clear. So here you have the digit tip kinematics, okay, and this is the smoothed motor unit discharge rate. So you have some motor units that are the coefficient of variation of the discharge rate is very flat, while some other units, the coefficient of variation is very high, and you can see that they perfectly and precede the kinematic. So this is because of the neuromechanical delay between neural activation and force activation. And this you can see across many times. So then if you take this motor unit, and this is very interesting for you, some of you that are interested in myocontrol, when you take this motor unit, you can, for each subject, this is the first correlation coefficient. This is a bivariate correlation. We can see that if you take this prime mover motor unit, you're basically estimating movement perfectly. So it's very important to find this unit. On the other hand, these units that are flat, they are not constructive movement. And this is very important because the mind control, so there are so many algorithms developed for mind control, and some of these work, some of these don't work, but there's a lot of problems with this algorithm. And one fundamental link from these studies shows that in the EMG, you are basically taking all of this. Yeah, so you're taking all of these units; they're all signed together, and then you have one signal that has it's basically what the British people say porridge. It's a porridge. Yeah, you have so much contribution there that is really not clear. So how can you estimate individual? You have either to decompose or you have to have very sophisticated AI because it's a complex problem here. You have a lot of information there that is summed, but EMG has that information. So it is, in my opinion, it is our duty to improve software and not to create new hardware. Because the hardware is already good. So I don't understand why we should have new hardware when it's a software problem. It's a mathematical problem, and I think so we have to do better, in my opinion, for the microcontroller. So now we have this data, and there has been a lot of hypotheses about that in the past. Desmet was one of the first that hypothesized that you have some different neural commands that can either project to the muscle that is doing the task, or they can also link to other muscles. And we also more recent study proposed this view, so that you have the nervous system will send one signal to multiple muscles. And there is one experiment that actually was designed by Desmet, and this experiment is very interesting because you can study this architecture by doing a very simple experiment where you have so this muscle, the first dorsal interosseous, has two actions. One is the abduction, so this movement here, and then this is a prime mover. This is the main muscle that does this. You don't have another muscle that can do that. It's only this muscle. Then this muscle can also do this. But this it's a synergist activity. So it's because you have the flexor of the index that is mainly pulling the tendon, and then by this muscle, if you do this movement, you see that the muscle is activated a lot, and this is supporting the joint. It's a synergistic muscle, and you can then what Dennett found that basically he hypothesized this, that when you have an abduction, there is some, whenever in this case it's a flexion, never when there is a flexion, you have some input that goes in the flexion that is leaked, so that we have this, the brain, the nervous system is not selectively controlling these muscles, but it's controlling the flexor, and some of the input is just going there. And this could be also due to alternate feedback integration. So we wanted to investigate that. So we used here the thin film electrodes that probably you've seen from publication of Professor Muscelli, that you have these thin film electrodes that they have a large number of channels, so they can give you a very precise estimation of the nerve movement. You can track motor units very well across tasks. With the surface, it's always a little bit problematic if you have changes in muscle structure. So what we did there, we did this experiment. So you can see here the implants. You have an implant in the first dorsal interosseous and also in the flexor. But we will just discuss about this today. And this is the implant, and then we also have the surface imaging sensor. And this then we found actually the surface imaging sensor can estimate it also quite well. We developed an isometric force sensor with a 3D axis load cell so that the subject can exploit the full degrees of freedom. You can see here the subject is changing with the hand fixed, and he can go to any place. And then the feedback was like always in a form of ramp movement. So they were seeing both of these plots, the subjects, so they could modulate either abduction or flexion in this case, very precisely at the exact force that we want. And then we wanted to see the motor units that are in one dimension against the other. So what we found was that across within the same force direction, if you do two times this run or 3 times within the same force direction, you find that the same motor units, each of these plots is the same motor unit, is recruited exactly at the same run as the same recruitment order. Okay, there is no violation in recruitment order. You can see here very strong correlation. On the other hand, when we track the motor unit, then we had so many units that violated it. Okay, so this does not mean that the size principle is violated. Because if you have the same input, the size principle still exists because it responds to the biophysics of the cell of the motor nerve. But what this means, it means that the central nervous system can go beyond the size principle, can selectively control enough occurrence these motor units. And you can see that there is a very strong violation of recruitment order. So the recruitment order is not fixed. Okay. And you can see a lot of that. So now we wanted to understand, this was already discovered by Desmet in a publication in Science in the 70s that has been ignored by quite the community for so many years. But this is, and he did that with the very old technology. But we found really very, very similar things. So then what was the next question was, why this is happening? Desmet could not find that because he was finding one motor unit at a time. But now we have a population view of the motor unit. So you can see here exactly what's happening. So you have an abduction task, and this is the flexion task. So this is the force, and this is the RMS of DMG. So in this, what we did here, we always normalize the force, not based on absolute force, but on EMG activity. Because when you do inflection, the force is much higher than abduction, because you have two muscles. So we wanted to have this exact same EMG activity to compare the two tasks. And you can see here during the abduction task, and this is the recruitment order that we always observe, that you have an increasing number of units. During the flexion, this is the same unit. You can see, for example, this unit here that was recruited very late. Okay, here, it was the first to be recruited. So then there was such a strong switch from motor units that are recruited. And interestingly, and this was an unexpected finding, is that even though if there is an inverse, the inverse relation between discharge rate and so the onion scheme This is something that's been called by Professor De Luca as the onion scheme, which is basically the inverse relation between discharge rate and recruitment threshold. So the motorists that are recruited early, they fire at higher rates. You can see that this is preserved. And this is very interesting because then the onion scheme is not dependent on the size of the unit, but it's rather dependent on the activation. So, and this has been hypothesized, actually, in one of the seminal works by De Luca that says that the common input, the common drive, is what defines the onion scheme. And that this is now experimentally true, this hypothesis. You can see even if we have a strong difference in recruitment threshold, the onion scheme is preserved. So the onion scheme is not due to intrinsic property of the motor neurons, but rather to common input. And this also means that probably we can train the onion scheme. Yeah? And it's something to estimate. But this also means that there are different inputs, because you have two different recruitment thresholds. The onion scheme is preserved. This means that the synaptic input to the currents that are going in this pool of motor neurons are different. And one way to estimate the difference is either, as I explained before, with the factor analysis, or there is this other method which is looking at the correlation in the frequency domain with coherence analysis, and this looks at the fluctuations of these components here across all the frequency spectrum. And if we do that, if we look during the abduction and inflection, you can see this is the coherence at different bands. So you have the low frequency bands, the afferent bands, and the more cortical high frequency bands that come from motor cortex that we can study. When we looked at this, we found no differences in the low frequency bands, in the tremor, in the alpha and delta, but very strong difference across all participants in the beta band. So it means that the signal that the cortical or afferent are transmitting is actually determined by different inputs, different synaptic inputs. And so these two tasks are different. So in a summary, the hand neural signatures are more complex than previously assumed, and the neural control of the human hand operates on a high-dimensional scale with only a few motor neurons in body-specific movement. So this then we can conclude that there are distinct common synaptic inputs that determine the difference in neural control across multiple force directions. And this brings me to the last part of the presentation, is how can all this information I've been talking translate into assisted neural rehab. So this was in 2020, we discovered that by using some electrodes, textile electrodes placed on the forearm of people with motor cortex paralysis. So this is the one participant from the brain trial that had a UTA array implanted in motor cortex. So these are people with C4, C5 tetraplegic, motor complete, motor and sensory complete spinal cord injury participants that have absolutely no movement in the hand. So what we found is that when we placed these high-density electrons in the forearm, we could estimate what the person was thinking, was attempting to do. And what we hypothesized is that there must be some neurons that are below the level of the injury that are still functioning. And because most spinal cord injury are due to a contusion, it's never a clear cut. The spinal cord is a traumatic accident, for example, with falling from a bike or a motorcycle. This contusion always leaves some spared connections. And these spared connections are very precious, what we found here, because we can tap into these spared neurons and let the patient see again that some movement that are lost, functionally lost, but not lost from an EMG perspective. And this is what we saw. We saw that when the patient was attempting movements, we would find some very clear motor units that were encoding that specific action. And you can see here a person that was attempting the movement. So now this was done 4 years later with state-of-the-art technology. This was then we went away from the textile and we used, in this case, you can see our Quattrocento system that we mapped all the forearm electrical activity. And by using state-of-the-art technology, we could find even more motor units. You can see here, this is a person with a C4 spinal cord injury and had a lot of motor units that were there. and we're encoding the movement very clearly, just like healthy individuals, as I showed you before. You can see here that there are motor units that are very well correlated with the digit kinematics. And you can see here, and then we collected 8 subjects, and you can see the number of motor units, it's very high in the motor complete persons. Then we developed a neurofeedback so that we had the 2 hands, one hand, because we cannot see the hand of the patient that is moving. So one hand we wanted to control, and then we asked the patient to map, to control this hand in real time with a machine learning method. And you can see that the person, in this case, a C4 spinal cord injury, can be able to switch between the digit flexion and the grasp. And then we wanted to also train these patients with real-time motivated feedback. And you can see this was a patient that after one month of training was able to very well selectively activate to match the force task that we had here. Now I show you the last part. This is we still all unpublished data because we have a startup, so that this will be soon accessible to all people in the world. Because the study will be published soon. But what I wanted to show you in the last slide is because now it's obvious that we have a signal that we can access non-invasively from the forearm muscles, and we can access this signal, and we can control, for example, prosthetic. So what we've been doing now for the last 3 years is to develop a new form of control. So something that there are so many exoskeletons available in the market, but there is nothing that is very light, that is high performance. That is an exoskeleton that is not like cumbersome. So we've been spending now also time to develop such system so that we have a control unit that can be controlled directly from the imaging feedback. And you can see a patient that had a spinal cord injury and could not move the hand for many, cannot move the hand at all. And we can now do a quite complex task. And so this means that these patients are far from being paralyzed. So they have a signal that we can interface in a non-invasive way. So with this, I close my talk. And if you have any questions, please go ahead.